So our last speaker is going to be Emily Filiberti, and she is a graduate student in the Department of Wildlife, Fisheries, and Conservation Biology at the University of Maine. Emily is working with Amber Roth, and her thesis focuses on estimating adult annual survival for golden-winged warblers throughout their breeding range. So Emily has kindly agreed to fill in for Amber today. So thanks for joining us, Emily. Take it away.
Sorry, I forgot to unmute. Can you guys hear me? Okay, so thank you guys for the opportunity to join you today. As mentioned, I'll be presenting on behalf of Amber Roth, and I'll be talking about, among other things, our efforts to improve estimation of golden wing warbler survival. So golden wings were assessed as being in high urgency of conservation attention and recovery. As you can see, their population has declined by about 60% since the start of the breeding bird survey. And their population is largely separated into two regional populations, one in the Appalachians and one in the Great Lakes, with only approximately 3 to 4% of the population occurring in the Appalachian region. So they're currently listed as threatened on the IUCN Red List, also on the PIF Red Watch List. They are threatened in Canada and a species of greatest conservation need in 27 of the state wildlife action plans. And they're also under review for ESA protection in the US. Potential contributing factors to their decline, not an inclusive list, habitat loss in both the breeding and wintering grounds, hybridization with blue-winged warblers, and also just general poor overwinter survival. So thanks to the many efforts of the Golden Wing Warbler Working Group members, golden wing recovery activities have been undertaken in all 4 phases at this point, with members conducting pretty extensive research throughout the full annual cycle. For example, golden wings have a monitoring program throughout the Appalachians, as well as site scale evaluation protocols to really assess implemented management actions. Additionally, a full annual cycle plan was completed in 2012 and partially revised in 2019 to incorporate new knowledge about the species and response to management. So because of this, we've undertaken more of a circular adaptive management process, meaning we continuously work in all 4 phases. However, I will mention that the social science aspect of golden wing recovery is one area where we really need to increase our focus to improve the conservation effectiveness of our activities. So on the R2R pyramid, our current project falls right in the middle as we try to identify limiting factors using vital rates and full annual cycle models. Given the severe global decline, it is critical that we understand the regional populations and how they are different demographically. And to do that, we need to dig into the underlying mechanisms of population growth rates. So there are several different mechanisms that drive population growth rates. Different demographic parameters all contribute to population growth or decline, with some contributing more than others. And understanding the relative importance of these different parameters across different regions is important in our understanding of why a population is either increasing or declining. And for a migratory species such as the golden wing, we have to consider outside the breeding grounds. So what's happening during migration and also on the wintering grounds? What does survival look like during these time periods? And understanding this can really help with current and future conservation strategies. So one approach to understanding the complexity of populations is through the use of integrated population models, or IPMs. And these are models that jointly analyze data on both population size and demographic data. So the structure of an IPM looks something like this diagram with 3 distinct categories of data: productivity, population count, and capture recapture. And from the capture recapture data, we can derive estimates of age and or sex specific survival, as well as breeding versus wintering versus migratory specific survival. And this really allows us to understand which components of survival have the biggest influence on population growth. So our current project, which is funded by US Fish and Wildlife, aims to construct a full annual cycle IPM for golden wings. And this is a pretty large collaborative effort, currently being led by Amber Roth's postdoc at UMaine, Jessica Hightower, and it includes data collected by many different collaborators. And within this IPM, the survival component is the focus of the R2R funded project. So survival estimates can be derived from band recaptured data or through telemetry. However, information can be quite limited for female golden wings, as they're pretty elusive and more difficult to capture than males, and they're also rarely re-encountered in subsequent years. So part of our project is deploying low-tech nanotags on both male and female golden wings across their breeding range in an effort to estimate sex-specific survival. So these tagged individuals can be detected by both the MODIS wildlife tracking system stations and with our handheld ground receivers. And as you can see on this map, the MODIS network is rapidly gaining in popularity with a recent explosion of stations throughout North America. And we were actually able to contribute to this network last spring in 2021 by constructing 2 stations near our study sites in Rhinelander, Wisconsin. So for our birds, we affix the low-tech nanotags to the back of both male and females using the leg loop harness method. And these tags are estimated to last approximately 400 days, which is long enough to detect returning tagged individuals in subsequent years. So last year, we were able to deploy 82 nanotags in 3 states, Wisconsin, Pennsylvania, and Tennessee. And we also installed 2 MODIS stations in each study area, so 6 total MODIS stations. As you can see, we didn't have as high of female representation in the Appalachians as we would have liked, but range-wide, we were able to deploy 31 nanotags on females and 51 nanotags on males. Some preliminary results from our field season this year. You can see our return rates this spring, which include both color-banded control males, so males without any tags, and tagged males and females. And again, we needed the control males just to assess possible negative tag effects. So you can see that tagged adult males had pretty similar return rates as our control males, which could suggest there wasn't an adverse tag effect. However, our tagged females returned at much lower rates, with none detected or recited in the Appalachian states, and only a couple detected in Wisconsin. So it is possible that the tags affect females differently than males. And I will mention, in an attempt to minimize tag effects, we did end up shortening the antenna length this season to the shortest possible length without losing substantial detection distance. So the R2R project objective, which was to expand nanotag deployment to improve apparent survival estimates at range-wide and regional scales. We were actually able to, with these resources, expand the study this year to include 6 additional states and 9 additional study sites. So with the addition of these collaborators, we were able to boost our 2022 Appalachian female sample size to 30 and our Great Lakes to 44. So we're pretty pleased with the number of females we were able to tag, given the difficulty in finding and capturing them, particularly in the Appalachians. And we look forward to returning all of us collaborators next year to find returning birds in 2023. So with that, I'll just mention funding for this project primarily comes from U.S. Fish and Wildlife, as well as a number of other funding sources. And I will shout out the Knobloch Family Foundation for being a primary funder. And I'd like to thank all of our NanoTag collaborators, as well as our project team members for making this expanded project possible.